Hey everyone, and welcome back for another deep dive. Today we're taking a bit of a turn from our usual focus on DNA, the bedrock of genetics and all that, and
Mhm.
diving into a world of tiny molecules with a huge impact.
Yeah, it's pretty amazing when you think about it. These molecules are like the puppet masters of the genetic world.
I like that analogy. It's like we're going behind the curtain to see how the magic trick is done, except in this case it's real science, and the illusions are actually groundbreaking discoveries. You guys did some really cool research on this. I'm really excited to dig into this with you.
Happy to be here.
So we've got two studies we're going to be diving into today. One takes us deep into the world of this tiny little worm called C. elegans, and the other explores the malaria parasite, which might not sound glamorous, but trust me, it's way more interesting than you might think.
It really is. Both of these studies focus on this fascinating phenomenon where these tiny RNA molecules, they're called microRNAs, act like genetic switches, basically turning genes on or off throughout an organism's life.
Okay, so before we get too far into the weeds here, can you break down why this is such a big deal? I mean, we always hear about DNA being the blueprint of life, right?
Right, and for the longest time, we thought RNA was just the messenger, carrying the instructions from DNA to make proteins. But what we're seeing now is that RNA is actually calling some of the shots. It's like this whole other layer of control we didn't even know existed.
Wow, so it's like RNA is the stage manager making sure the play goes off without a hitch.
Precisely. And the first study really highlights this. It focuses on a specific microRNA called let-7 in our little worm friend C. elegans.
Okay, so why C. elegans? What makes this tiny worm such a good candidate for studying something as complex as gene regulation?
C. elegans is kind of like the lab rat of the worm world. It's tiny, transparent, and its development is so well studied. It's like we have a genetic instruction manual right in front of us. Plus, they have these genes called heterochronic genes, which are basically the timekeepers of development.
Heterochronic genes. Are we talking
Yeah.
about time travel and worms now?
Haha. Not quite time travel, but it's a fun way to think about it. Basically, these genes make sure that everything in the worm's development happens in the right order at the right time. They're like the conductors of an orchestra, making sure the violins come in at just the right moment. And what the researchers found is that messing with let-7 is like messing with the conductor's score.
Okay, now I'm really intrigued. Tell me more. What happens when this let-7 microRNA gets disrupted?
So in normal C. elegans development, let-7 levels increase right when the worm is supposed to transition from its larval stage to adulthood. But when the researchers messed with let-7 prevented it from doing its job, the worms got stuck. They just kept molting into bigger larvae, never really growing up.
Wait, hold on. Are you saying these worms were basically stuck in a perpetual childhood, like Peter Pan, but with worms?
Exactly. Imagine a caterpillar just getting bigger and bigger, but never forming a chrysalis. It was like someone hit the repeat button on a specific stage of their life cycle.
That's both amazing and a little creepy. But how did the researchers even figure out that this tiny RNA molecule was the culprit behind this developmental freeze frame? It's not like they could just ask the worms what was going on.
Uh-huh, right. They had to get pretty creative with their techniques. They used some pretty powerful tools like northern blots and S1 nuclease mapping.
Okay, I'm going to need you to break those down for me a bit. I'm picturing some seriously high-tech detective work here, but I'm not sure I understand the tools.
Imagine trying to find a single typo in a library full of books. That's how precise these techniques had to be. They basically allowed the researchers to isolate and study let-7, which is incredibly small, and then track how its presence changed throughout the worm's development.
Okay, so some serious scientific sleuthing to prove that let-7 was the mastermind behind this whole developmental drama. But how does this tiny RNA molecule actually exert so much control? Does it just like yell at the other genes to stop growing up?
Haha, that would be something to see. But it's a bit more elegant than that. But let-7 actually works by binding to messenger RNA, which is the molecule that carries the instructions from DNA to build proteins. By binding to specific mRNAs, let-7 can either block them from being translated into proteins or even target them for destruction.
So it's like let-7 is the ultimate editor for the book of life, deciding which chapters get read and which ones get tossed out.
That's a great way to put it. And one of its favorite targets is a gene called lin41, which is essential for the worm to transition to adulthood. Let-7 basically puts lin41 on hold until the worm is actually ready to grow up.
Wow, this tiny molecule is basically calling the shots for these worms' entire lives. But before we get completely lost in the world of microscopic worm drama you mentioned, we're also diving into some malaria research today.
Yeah.
What does worm RNA have to do with battling this devastating disease? So we've seen how these tiny RNAs can basically orchestrate an entire developmental saga in a worm, but what about in something like a deadly parasite? This next study really blew my mind because it shows just how adaptable these microscopic villains can be.
Yeah, absolutely. And this is where things get really interesting from a global health perspective.
None.
The second study you shared focuses on the malaria parasite and this gene called PFMDR1, which plays a key role in drug resistance.
And drug resistance is a huge problem in the fight against malaria. It's like every time we develop a new weapon, the parasite just pulls out a new shield.
It's a constant arms race, for sure. And PFMDR1 is a powerful weapon in the parasite's arsenal. It basically codes for this protein that acts like a gatekeeper on the surface of the parasite cells, controlling what can get in and out.
So if the parasite can control what comes in, that means it can potentially block things like antimalarial drugs from ever reaching their target.
Exactly. Imagine a fortress with a really, really picky guard at the drawbridge. That's PFMDR1 in a nutshell. And what's even more concerning is that it only takes small changes, sometimes even a single mutation in PFMDR1, to make the parasite resistant to multiple drugs.
Oh, wow. So we're not just talking about resistance to one drug. We're talking about a parasite that could potentially outmaneuver an entire arsenal of treatments. So how did the researchers even begin to untangle this PFMDR1 puzzle?
Well, they used a really clever approach. They compared two different strains of the malaria parasite side by side. One strain was sensitive to the drug chloroquine, and the other was resistant.
Okay, so they have their control group and their experimental group, both with different responses to this drug. What did they do with them?
They actually went in and specifically manipulated the PFMDR1 gene in both strains. This allowed them to isolate the effects of mutations in that gene on drug resistance. It's like rewinding the evolutionary clock to see exactly when and how the resistance popped up.
That's fascinating. So they were basically looking for the smoking gun, the precise genetic changes that allowed the parasite to shake off the drug's effects. So what did they find? Was it as simple as this mutation equals resistance?
I wish it were that easy. It's rarely ever that straightforward, unfortunately. While they found that PFMDR1 mutations definitely play a huge role in drug resistance, it's not always a one-to-one relationship. The impact of a particular mutation can actually vary depending on other factors like the genetic background of that specific parasite strain.
So it's a complex puzzle where PFMDR1 is just one piece, albeit a crucial one. What are some of the other factors that came into play?
Think of it like this. Even with the same faulty drawbridge, some fortresses might have thicker walls or a wider moat. In the case of the malaria parasite, these other factors could be additional genes that offer backup resistance mechanisms or even differences in the parasite's metabolism that affect how it processes the drug.
It really highlights how adaptable these organisms are. They're constantly evolving and finding new ways to outsmart our best defenses. This is some next-level biological warfare we're talking about. But we've thrown a lot of complex science at the listeners today. What's the key takeaway you want them to walk away with?
That's a good question. I think the biggest takeaway here is that the more we study these tiny RNA molecules and their influence on things like development and drug resistance, the more we realize how much we still don't know. We're constantly uncovering new layers of complexity, and it really underscores just how essential basic research is in all of this.
That's like the deeper we dive into the microscopic world, the vaster it becomes.
Yeah.
But that's also what makes it so exciting. There's always something new to discover. It really is like peeling back the layers of an onion, except instead of making us cry, it's giving us all these fascinating insights into how genes are regulated.
Exactly.
And potentially how we can use that knowledge to improve human health.
Absolutely. And it makes you wonder what else is out there. What other secrets are these RNA molecules hiding? What haven't we even begun to uncover yet?
I know. It's both exciting and a little daunting, right? Like we keep thinking we've got a handle on things, and then boom, a whole new layer of complexity pops up.
That's the beauty of science, though, isn't it? There's always something new to learn. The more we study these tiny RNA molecules, the more we realize how much they impact our understanding of everything from disease to evolution.
It's really incredible when you think about it, these tiny molecules holding the keys to some of the biggest mysteries in biology. Well, we've covered a lot of ground today, from worm development gone haywire to the malaria parasite's arsenal of defenses. Is there any final thought you want to leave our listeners with?
Maybe just a reminder that even though the research can seem really complex, it all comes back to these fundamental processes of life. And who knows, maybe one day understanding how a tiny RNA molecule controls a worm's development could lead to breakthroughs in treating human diseases.
Or even reversing aging. Okay, now that's a thought to ponder. It just goes to show you sometimes the smallest things can have the biggest impact. If you're as fascinated by these tiny RNAs as we are, be sure to check out the show notes, where we'll have links to the original research so you can dive even deeper into these incredible studies And as always, we want to hear from you. What blew your mind the most? Head over to our website or find us on social media and let us know. Until next time, keep those brains buzzing and stay curious.